This tutorial is all about gas exchange surfaces and specifically within the human lungs. On the screen in front of you, you should see the exam question that says, how do the tissues in the gas exchange system contribute to the functioning of the lungs? Now, there's a whole range of different tissue types that you need to be aware of. In fact, there's sort of 5 main ones, but within the mark scheme for this particular question, we're listed 7 different tissue types. The first is the goblet cells, and the goblet cells are also shown on the images on the slide. Goblet cells in the lining of the airways secrete mucus. And the mucus traps microorganisms and dust particles in the inhaled air, stopping them from reaching the alveoli. Now, a second tissue that you should be aware of are the cilia, also labelled on the diagram there. Cilia on the surface of cells lining the airways beat the mucus. This moves the mucus, plus the trapped microorganisms and dust, upwards away from the alveoli towards the throat, where it's then swallowed. And this helps prevent lung infections. Not labelled on the diagram, but another tissue you should be aware of and we'll look at today are elastic fibres. Now, elastic fibres in the walls of the trachea, bronchi, bronchioles, and the alveoli help the process of breathing out, exhalation. On breathing in, inhalation, the lungs inflate and the elastic fibres are stretched. And then the fibres recoil to help push the air out when exhaling. A fourth tissue type you need to be aware of are the smooth muscle. Smooth muscle in the walls of the trachea, bronchi, and bronchioles allow their diameter to be controlled. During exercise, the smooth muscles relax, making the tubes wider. This means there's less resistance to airflow and air can move in and out of the lungs more easily. A final one I'll mention for now is the rings of cartilage in the walls of the trachea and the bronchi, which are there to provide support. So they're strong but flexible. It stops the trachea and the bronchi collapsing when you breathe in and stops them collapsing as the pressure drops. Now, projected onto the screen is the nasal cavity. You need to be aware of the key features of the nasal cavity, as well as each of their functions. So the first key feature you need to be aware of is the fact that it has a really good blood supply over a large surface area. This enables air to be sort of warmed. It ensures the air is warmed to the body's temperature to protect the delicate lung tissue. They also have a hairy lining that secretes mucus to trap dust and bacteria to prevent lung tissue from irritating and infecting the lungs. And they also have a moist lining, which increases humidity of incoming air to reduce evaporation from exchange surfaces. Now, I'm going to give you a moment to have a look at the slide in front of you. There are 2 tubes shown on the slide. Which one do you think represents the trachea and which one do you think represents the esophagus? The trachea is on the left-hand side. The reason why we can tell that is the trachea has cartilage to keep the airways open, preventing collapse as pressure changes. In comparison, on the right-hand side, the tube on the right, that's the esophagus. And you can tell because it's got infolding, so it can stretch as food passes down. Again, you need to know about the trachea. You need to know about its key structures and the function of each. So the trachea has goblet cells, it's got cilia, it's got cartilage, specifically it's C-shaped cartilage, and it's got elastic fibres. So the goblet cells in the trachea are going to help release mucus to trap pathogens and dust, the cilia to waft the mucus up to the airways for it to be swallowed, the cartilage to provide that support preventing collapse as we have the pressure change, and the cartilage is C-shaped, which allows flexibility and space for food to pass down the esophagus. And the elastic fibres help in expiration as they recoil, helping to decrease volume and push air out. This slide just shows the esophagus and trachea again. This time you can see the bulge where the food is passing down the esophagus, which is just sort of demonstrating the importance of having the C shapes of those cartilage. If the cartilaginous rings were complete, you wouldn't then have that space to accommodate for the food passing down. Now this slide here looks at the bronchus and the bronchioles. The bronchus has a similar structure to the trachea. So when you go down the trachea, it branches off into two branches, or two bronchi. So each bronchus has similar structure to the trachea, but instead of C-shaped cartilage, it actually has pieces of cartilage, and they're labelled on the diagram on the left-hand side. The bronchioles on the right-hand side, the largest, are similar to the bronchus, but the smallest bronchioles leading to the alveoli have no cartilage, and they sort of have flattened or squamous epithelium to allow for gas exchange. So here we have an image of that squamish flattened epithelium on the right-hand side, and an image of the alveoli on the left-hand side. So the alveoli are very small. They are sort of 100 to 300 nanometers across, but there's hundreds of them per lung. The surface area of the exchange system, so taking into account all the alveoli, is estimated to be about 70 meters squared, which, considering how small each individual alveoli is, that is pretty extraordinary. So the squamous, the flattened or pavement epithelium, this type of epithelium lines the alveoli and the smallest bronchioles. It's permeable tissue. It provides a very thin layer for oxygen and carbon dioxide to diffuse through. And it's lined with a thin layer of moisture that aids diffusion. As well as these structures, you should also be familiar with Fick's law. Now, hopefully you came across Fick's law at GCSE, but if you don't know it, it's that the rate of diffusion is inversely proportional to the surface area times the difference in concentration divided by the thickness of the exchange surface. So what this sort of law tells us is that diffusion is going to be most efficient when there's a large surface area, a small distance for diffusion across a partially permeable membrane, and a really steep concentration gradient. Our ventilation systems, or ventilation in itself, ensures there is a high concentration of oxygen inside the alveoli and a low concentration of carbon dioxide. An efficient blood flow ensures a high concentration of carbon dioxide and low concentration of oxygen in the capillaries, and hence a steep concentration gradient between the alveoli and the capillaries, ensuring the efficiency of our exchange surfaces. On the slide now is a past paper question. This would have been worth about 6 marks. And it says, why do you think smokers get more infections of the breathing system than non-smokers? I'm just going to give you a moment to think about that. Now, the reason why smokers get more infections of the breathing system than non-smokers is because the trachea is lined with ciliated epithelium and goblet cells that secrete mucus. The mucus, as we've already mentioned, traps dust and bacteria. And the cilia are designed to move the mucus and trap particles from the lungs to be swallowed and digested. Now, in smokers, the cilia becomes damaged so that it can't beat. The mucus with the bacteria therefore stays trapped inside the lungs, and more pathogens will reach the lungs. So smokers are more likely to get infections of the breathing system than non-smokers with active cilia. You also need to be aware of the condition of asthma. Obviously quite common. Triggers of asthma include house dust mites, cigarette smoke, pollution, pollen, and even stress. During an attack, cells lining the bronchioles release histamines. Those histamines cause inflammation of epithelial tissues, stimulate the goblet cells to produce excess mucus, and cause the smooth muscle in the bronchiole walls to contract. The airways narrow and fill with mucus, making it really difficult to breathe. Some asthma medication is taken every day, usually steroids, which reduce the sensitivity of the lining of airways to asthma triggers. And then this reduces the likelihood of an attack in the first place. We're going to go through just sort of one or two more questions to finish off now. So here we have two images. They're showing sections of lung tissue. The one on the left-hand side is from a non-smoker, figure 2.2.A. And the one on the right-hand side is from a smoker who suffers from another condition called emphysema. How does lung tissue differ in these two individuals, and what are the consequences of these differences to gas exchange? I'm going to give you a moment to think about that. So in the image on the right-hand side, that's the one from a smoker, you can see that we've got ruptured and collapsed alveoli, i.e. we have fewer alveoli. This means we have larger air spaces, but smaller surface area. As a result, gas exchange is going to be less efficient as the rate of diffusion is slowed due to the smaller surface for exchange to take place. Another question here. So the question is, why do people suffering from emphysema have difficulty in expelling air out of their lungs when they breathe? Again, I'm going to give you a moment to think about this. Now, the reason is because the alveoli can't recoil due to the loss of elastic tissue in those suffering from emphysema. The elastic tissue is actually destroyed by phagocytes.